In this segment, we are going to discuss about restriction endonuclease enzyme. Endonuclease enzymes are those enzymes which digest the DNA or cuts the DNA at a specific sites, and these specific sites are called as restriction sites or restriction sequences. So cuts the DNA at a specific sites called as restriction sites or restriction sequences. In the year 1968, the first restriction endonuclease was isolated from Haemophilus influenza bacterium, and the restriction endonuclease name is HIND2. In 1968, first restriction endonuclease was isolated from a bacterium which is Haemophilus influenzae, and the restriction endonuclease was HIND2 restriction endonuclease. Moreover, around 900 restriction endonucleases has been isolated from over 230 strains of bacteria. Right? Now discussing about the types of restriction endonucleases. So basically, majorly there are three different types of REs, that is restriction endonucleases: type 1 restriction endonuclease, type 2 restriction endonuclease, and type 3 restriction endonuclease. So type 1, type 2 and type 3 restriction endonuclease. So first is type 1 RE. Basically, the job or the function of this type 1 RE is undergo restriction endonuclease, that is it undergo digestion as well as methylation. So role is to perform the restriction function. When I am using the word restriction function, that means it is cutting the DNA at a specific base pair sequence, and the sequence is called as a restriction sequence or restriction sites. So the job of type 1 RE, that is type 1 restriction endonuclease, is restriction and methylation, and we all know what is methylation. It recognizes of the restriction sequences of 15 base pair. Recognition sequence is of 15 base pair, and it cut the DNA at around 1000 base pairs away from the 5' end of the DNA. So it cuts the DNA about 1000 base pair away from 5' end. That means, for example, if this is the 5' end of the DNA, this is the 3' end of the DNA, right? Only one strand, one template we are talking about. So type 1 RE cuts the DNA about 1000 base pairs away from the 5' end. That's why such restriction endonucleases are not used in our DNA technology. So they are not used in our DNA technology, that is restriction or recombinant DNA technology. The second type of restriction endonuclease is type 2 restriction endonuclease. Type 2 restriction endonuclease. Now, recognition sequence is about 4 to 8 base pairs only. So, at a shorter sequence, recognition sequence is about 4 to 8 base pair only, and it cuts only methylated sites, right? And the cut is also very specific. specific. That means within the recognition sequence it is cutting. Say for example, if the sequence is of 4 to 8 base pairs, let us assume 6 base pairs. Right? An average of 6 base pairs is a recognition sequence for the type 2 RE. Then type 2 RE cuts where? Exactly or within the recognition site only. In case of type 1 RE, it is cutting 1000 base pairs away from the 5' end. So this is used. So it cuts at methylated sites within restriction sequence only. So if it is cutting within the restriction site of the sequence, this is used extensively in rDNA technology. The third type of RE is basically called as type 3 RE. It is type 3 RE. Type 3 RE shows the functions as well as properties of type 1 and type 2. That is, it is between type 1 and type 2 restriction endonuclease. One more point regarding type 2 RE, that around 350 REs of type 2 are known or has been isolated till date. Now, talking about the mechanism of action of this restriction endonucleases, we will take few examples. The example that we are going to take is the example of EcoR1, right? But before going into the details of the mechanism of this restriction endonuclease, let us first understand like nomenclature, how to nomenclate these restriction endonuclease enzymes, right? If you remember in the chapter 2 of 11th biology, there was a chapter 1, binomial nomenclature, right? The certain norms and the rules of nomenclature was given, the generic name, specific epithet, like that. The same rules we have to use, or the rules are given in order to nomenclate restriction endonucleases. So let us discuss the rules of nomenclature, that is nomenclature of restriction endonuclease enzyme, and then we will discuss the mechanism of action. So nomenclature of restriction endonucleases. 4 to 5 rules are given as a part of nomenclature of restriction endonuclease. The first is the name of enzyme is derived from the name of the bacterium or the prokaryotic cell from which it is isolated. So first rule is name of enzyme is derived from the name of prokaryotic organism from which it is isolated. Let us say the prokaryotic organism is bacterium, right? We will discuss one example after discussing the rules of nomenclature of RE. Now, first letter of enzyme will be the first letter of the genus of this prokaryotic organism. So first letter of enzyme will be the first letter of genus, right? Then the third rule is the second and third letter of enzyme is equals to the first two letters of species, right? This is very interesting to note and it is important also. Second and third letter of enzyme will be equals to the first and or the first two letters of species. first letter is equals to the first letter of genus only. Second and third letter of the enzyme is equals to first and second letter of species, that is specific epithet. And the fourth is first, second and third letter. All these things must be written in italics. So all three letters must be written in italics. Then fourth letter of the enzyme is equals to the first letter of strain. Fourth letter of enzyme is equals to first letter of strain, right? And this is also written in capital. Roman number at the end of the enzyme indicate the order in which enzyme has been isolated. So Roman letter at the end, like when we write the name of the entire restriction endonuclease enzyme, we generally write the Roman letter. So Roman letter at the end indicates the order in which enzyme was isolated from prokaryotic bacterium, right? So all these are the rules of nomenclating the restriction endonuclease enzyme. So let us take one example in order to understand how to nomenclate a typical restriction endonuclease enzyme, right? So the rules are written here. I will read once again the rules and we will see with the help of one example also. So we are taking the example as EcoR1 is a restriction endonuclease enzyme isolated from E. coli RY13 strain of the bacterium, right? EcoR1 is the name of the restriction endonuclease and E. coli RY13 strain is the name of the bacterium from which this EcoR1 was isolated, right? So first rule is name of an enzyme is derived from the name of the prokaryotic organism, right? E. coli, eco, simple. Second is first letter of the enzyme will be the first letter of the genus of this bacterium. Here Restriction and coli. This is genus and this is species. Escherichia genus and coli species. Generic name and the specific epithet of the specific name of the bacterium. So first letter of the enzyme will be first letter of the genus. What is the first letter of genus? E. So E will be the first letter of enzyme, right? Third rule is second and third letter of enzyme will be first two letters of species. First two letters of species are coli, C and O. So we have to focus on first two letters of species only. And these two first two letters of the species will be second and third letter of enzyme, because this is the first letter of enzyme. Second and third letter of enzyme will be equals to first and second letter of species. So CO, right? All three letters must be written in italics. All three letters must be written in italics. Right? Fourth letter of enzyme is equals to the first letter of the strain. Right? Now, this is generic name, this is specific epithet. RY13 is the name of the strain. So, fourth letter of enzyme, first, second, third. Now, fourth letter of enzyme is equals to the first letter of the strain. first letter of string is R, so we will write it as R, right? Roman letter at the end indicates the order in which enzyme was isolated from prokaryotic bacterium. So here it is 1, that means for the very first time EcoR1 enzyme is isolated from E. coli RY13 bacterium strain, right? So this is all about the nomenclature of a typical restriction endonuclease enzyme. In the next part we will discuss about the mechanism of action of restriction endonuclease enzymes.